do some glowing plants, y'all. This is a really cool form of technology. And here is the paper, and this is the first of its kind. So I actually think that you're going to find this to be a cool story. So I hope many of y'all do find this really cool. What the scientists were trying to do was to make what is called a biosensor. So a biosensor allows you to track something in real time in an organism. So similar to the fluorescence that we've talked about, where if you have a gene that's turned on in an animal, you tie on a glowing molecule. So that the protein, if it's in the animal, you've hooked on now a glowing piece of DNA, and that glow then means that whenever the protein pops up, you can see through the animal that there is a glow to it. That's really, really cool. Where it gets even cooler than that is if you don't just hook it to a protein, but you hook it to the RNA. So we kind of talked about this a little bit this past week, that it's not amount of RNA doesn't always translate to amount of protein. So if you've got a ton of an RNA molecule, you might not have the same amount of protein at the end of the day. Or maybe it's a good indicator of how much protein that you're going to have. But because the dynamics of the RNA will influence how much protein you make. Yeah, for a particular sequence, exactly, Jay. So not just universal presence or absence of RNA, but if you want to look for a particular kind of RNA molecule that will lead to a particular type of protein, and you want the RNA molecule itself to be visible. Why would you want to do this, Jay? So it's a way to visibly track changes in RNA and therefore changes in gene expression in real time, right? So you can imagine a scenario where you're looking at a plant and you put it into different soil, and the soil that you move it to has higher levels of iron. And you want to figure out if you've identified a gene in a way where can you see in real time this gene becoming activated? Or it could be the opposite, a gene that's already activated. So you see green, you see this fluorescent green signal, but then if it's something that needs to decrease, when in that type of soil, you lose that chemical signal. You lose the green, right? So those are the two sides of the equation that you can look for here. So it is a means to measure RNA now inside of plants that result in something that's visible, where you do not have to wreck the plant to see it. Exactly, real time. Now, Jay, the only catch is that you have to, it's not universal for every gene, right? You have to already have a target. You have to know or have an idea of what gene you're interested in. So this can't just be used for willy-nilly discovery. I guess it could be, but it would be very taxing, right? If you hooked this technology onto a ton of different RNA molecules and saw what glowed, it would be a huge range of things if you didn't know if you're what you were especially looking for. It would not. The reason for that, Jay, is because RNA is weird. RNA is weird, okay? And the reason for that, Jay, is because you can balance RNA molecules to make them last longer. And by that, I mean RNA degrades at different rates. So if your RNA molecule, it can be copied multiple times to make a protein, right? So translated. So I have, taking from my textbook, my DNA, a page, I've copied it, that's your RNA, and I'm going to read this recipe to make my bread. I can read that recipe page a couple of times and make bread a couple of times from this recipe. Sometimes. Other times, Jay, you can only read it once because it's put into the shredder after reading it one time. And so that gets to be a difficulty so that means that the amount of RNA that's in a system doesn't always correspond to the amount of protein. Because you can imagine a scenario, you have a few RNA molecules, right? And those few RNA molecules are enough to make a lot of protein, because that you can read those chapters a lot of times, right? You can use this page to make up bread over and over again, right? So that doesn't necessarily mean it's not a 1-to-1 ratio of one RNA molecule to one protein. There, it's few RNA molecules to a lot of protein. And then you can have the opposite too, where the RNA is modified in a different way that after it's been translated once, it gets targeted for breaking down. And that affects turnover rates of these things. So you would need a different tool. This would tell you changes in gene expression. You would have to do further validation to fully convince yourself and a reviewer that the protein levels are also changing. And that is relatively new. I would say that's in the past decade that's become a more commonplace. So yeah, you'll be measuring gene expression in real time. Is that when the end of the RNA turns double-stranded? Sometimes. Other times, Jay. So at the end of an RNA molecule that's going to be translated, it has what is called a poly A tail, which means it attached to the end of the RNA is a long series of A's. And remember, the base pairs that you use for DNA and RNA are AT or AU, right, for RNA, and GC. And so the end of the RNA molecule is just a string of A's. That is called the poly A tail. That is in part responsible for how long the RNA molecule can survive. If you rid yourself of the poly A tail, if you chop it off, then there's machinery inside the cell that says, oh, we don't need this anymore, let's chop it up. Yep, exactly. Damn, Karen, like it's written out like that. The sequence end of the RNA that's gonna tune a protein, and it's not the only mechanism, but it's a very common one, is this poly A tail of and it just goes, which is cool, right? And so tail and the length of that tail will influence how long that molecule lasts. You can imagine, Jay, if it's just a couple of A's, that it's like on a ticking clock, you'll lose those A's faster because there's less of them, which means you're going to degrade that RNA a little bit faster. Versus if you have a ton of A's at the end of it, then you have a higher likelihood of it being able to survive. So yeah, that's one means. That's not the only means of an RNA molecule surviving for a long time. That rule goes out the window if you've got a microRNA, because they're micros, so they're really small to begin with, so there's not a poly A tail. It has a totally different mechanism. Of course it does, because that's how this stuff works, right? But that would be the yeah, and exactly. It has like a degradation rate. It's like, you start a timer, and for the amount of A's that you have on there, that will correlate with how much time you have on the timer when you start it. And that is different based on what gene that you're doing it on. Like some of them are designed like to have fast turnover because you need to have a rapid response. And you don't always need them on, and you don't want them hanging around for a long time. Others, like what are called housekeeping genes, right? Those are always needed. Like those are always on. Those are things that keep your cells functional. And so so the process is just homeostasis. So to keep everything balanced. And so for those genes that are involved in this housekeeping process to make sure everything's functioning as it should, those have a long A because there's not a situation where you don't want that. So for example, things like the cytoskeletal matrix, the thing that keeps the cell's shape, that you're going to need, right? You don't want the cell to implode on itself. So you're going to have that. So that's a great question, Jay. You amazingly, I love do stuff at this point. You should know that I love the absurd, for better or for worse. I'm a big fan of the absurd here. So Jay is asking the question here, y'all, and I think it's important: is how do you know what to connect? What is the system? Like, what are you hooking on this green fluorescent protein to? What is the RNA molecule that's important? That's a very good question, Jay. Right? Because there's thousands of RNA molecules that you can choose. How do you choose it? The way they did it here was there already was a candidate list, meaning that what you can do, you can take a plant and move it from one type of soil to another, and then it's going to have changes in gene expression. Let's say after 24 hours, Jay, you take the plant out and you take out some cells and you put it in the sequencer and you figure out and you see there's differences. And that is your candidate list. That is your candidate list. Okay, the RNA sequence, yeah. So you do experiments that gets you a list. After that candidate list has been established, that's when you use this technology. That's a really, really good question. So it's doing a sequencing experiment to get you your candidate list, and then you do genetic engineering on the plant to hook on this green fluorescent protein to be attached to the RNA molecules of interest to you. Again, I don't think that it would work, Jay, if you didn't already have that list. You could, in theory, start just hitting random genes, like going to a database that tells you, like, hey, here's a list of genes in this plant, and then you just hitting every single one of them. But that would take an absurd amount of time. And also, you have no idea how you would activate those genes. I guess if someone made the resource, like if there was an online repository where you could just order and every single gene has been hit up with this glowing one. So if you were making it as a tool, that would be the time that you would do that. because otherwise, right, you presumably have a question, right? Like you're asking a question like, how does X affect Y? And so if you've made all these genetic changes, presumably you already know certain ones that wouldn't be involved because it's not like in iron processing, like if the soil levels of iron are different, right? So it would just, you starting off with a list, unless again, like you're an open access wackaloon that made these tools for other people to use, and then you would do everything. But that depends on the funding as well. This was actually done at Oak Ridge National Laboratory. So this, I believe, is in Knoxville, Tennessee. It's actually they have some secret like military level stuff that they do there as well. So yeah, all of it. So it already has been in animals, Jay. It just had never been done in plants. That was the catch. In animals, it has been established to work. No one could get it to work in a plant. And so this is the first instance of it actually working within a plant, which I think is really, really cool. It's weird to think that it did not work in another organism, but the reason for this was because what molecules they needed to do, right? So what they call this the glowy rabbits, for example. Now, the glowy rabbits to begin with, Dame Karen, were just GFP, so green fluorescent protein just turned on in the rabbit. And that wasn't a biosensor, right? So that is just you're turning on green for the sake of turning on green. Since then, they've also made biosensors in animals where, again, you'll only see that green turned on when it's a certain gene that's turned on. So, but the original glowing bunnies were just, let's turn on green fluorescent protein in the entire body of this animal to make it green. So that would be the difference. Like, that's not giving you a monitored output. Like, if you measure the fluorescence in the animal, right, it's only giving you data on that it is in fact, like, you can translate the amount of glow as it's how it's linked to a gene level, right? And that's only going to happen if you've linked it to a gene to be able to measure something. In the glowing bunny, it was just, we just want to make it green to make it green. Yeah. Thought that they didn't try plants before animals. We don't know. So I don't actually know based on this paper do stuff of like which one has been tried longer. It's possible that the issue is do stuff. Failed things don't get published because it's not considered sexy to publish something that's failed. Who's the GF? Yep, exactly. So glued, they did it for a couple of different reasons. One is that it's easily identified. Two, it's that it had been done in fruit flies and yeast and nematodes. And then it was just going for like, let's make it as complex of a mammal as we can. And so what they did was, it's not just turning on this green fluorescent protein or GFP everywhere. It's they are making a different protein glow. So the glow here that they're making is called a histone. Histones are the beads on DNA that are tied around the DNA to keep it small and compact. Every cell in your body and the rabbit's body has histones. And so what they did was they made the histones glow green, and the rabbit glows green because every cell of it in it has histones, and therefore it glows green. Does that make sense? So it's not that there's just this green molecule randomly floating around in the bunny. You've made a protein that it has in every cell glow green, and that's why it glows green everywhere. Otherwise, that glowing protein might end up being destroyed in the body of the animal because the immune system would probably be like, why we got this, y'all? Let's get rid of it. But if it's attached to something that's really important, then the body's like, well, this is important, so it's fine. And so that's like the policing mechanism of it. So the reason this took so long to do is because what they needed to do, again, is to mark the RNA. It was difficult to do. Protein is much easier. And it's in live. They want this biosensor to work in live plants. Normally, the way that you can mark RNA is that you have to kill the plant. It takes a lot of time to do cross sections and analyze tissue. The whole point of this paper is to show that scientists can look at these plants and in real time see what genes are changing under different environmental conditions. And those environmental conditions could be differences in soil, drought, disease state of the plant. So you can imagine a situation like, Jay, if we find a gene that protects against drought, like, oh, look, this gene got cranked up a little bit during drought conditions. Okay, what if I increase it even more artificially? Oh, look, now it's even better at surviving droughts and it still produces food for us, right? That's kind of what you're trying to go for here, is can you make things grow better in climates and in different ecosystems with a changing climate to keep feeding people? That's like their big takeaway there, and probably other stuff too, right? Like you can imagine a lot of different scenarios that this can work in. So the way the system works is that there is an RNA molecule. So do stuff, you can have it only active in certain parts of the plant. So if you have it only active in the leaves and not in the fruit, you won't even eat it, right? You would not eat the fruit that has it. But honestly, there would be no result. You would have no changes in it, because if it's not even the protein that glows, it's an RNA molecule, and when we eat fruit, that it's broken down in you. You're not gonna be like glowing as a result of it. Bigger things that happen to stuff are already like secondary metabolites that are in fruits and vegetables. For example, do stuff, if you gorge yourself on tomato juice and carrots, because of the secondary metabolites in those two food items, you can turn orange, right? So and that's without any genetic modification at all. That's just you can change yourself to be orange by just changing your diet, and you will not glow green. You'll not grow green from it. It would probably, coffee, have to be labeled as a GMO. If, Dame Karen, if it's in every cell of the plant, then technically, you've done an injection to change the DNA of the entirety of the plant, even if it is only activated in a subset of the plant. So this is one of those things where I'm just like, I just don't know where they would go with the definition of this, because it could be one that I could see going either way on it. But so what did they actually end up doing in terms of the technology? Because I know that's what Mrs. Spites is ready to be asking. So, Jay, there is a molecule that's called a ribozyme. I know, Jay. Excuse me now? It's called a ribozyme. So a ribozyme is a molecule that acts as an enzyme, which helps chop up RNA inside of the cell. Remember the alternative splicing that we talked about a few days ago, where you can take an RNA molecule and you can chop out parts of it and make a new combination to make it a different protein? The thing that helps do that is called a ribozyme. And so what they actually did here is quite clever. So the ribozyme that they utilized in this was to make it so that instead of splitting just a random piece of RNA and separating them out, what they ended up doing was they attached the ribozyme pieces together to pull together two components. Okay, so there's actually a really nice figure, I think. Here we go. Okay, so Jay, I think this is a genius method. You turn on a gene, okay? Your gene of interest. Normally, your gene of interest doesn't glow, right? Let's say that it is a gene that responds to iron deficiency in the soil. Okay, it turns on. So, Jay, how do we make that turned on gene green? In comes the ribozyme. So they chemically modified this enzyme to pull together two strands of RNA. So what they designed it for is that this side of the ribozyme is targeting a sequence for green fluorescent protein, or GFP. This side of the ribozyme, the other side, is targeting whatever gene that you're interested in. And what does the ribozyme do? It connects them together. And then the ribozyme floats away. And now you have an mRNA sequence that has part of it coding for green and the other part of it coding for whatever that gene was that you're interested in. They're literally just putting in a step to connect two things. Exactly. You are custom alternative splicing. Except here, it's just you're adding on at the RNA level this green put. And so that's giving you that direct look of, okay, how much of this is actually like on the RNA level. And here's how you can tell. So this is the process by which this works. And then when you code for it too, the ribozyme is part of that code here. And so when that ribozyme also is made and pulls these two together, then it excises its own self. And so they made a bunch of different variants of it in order to chemically prove that it can work in as one construct. So the idea of this is, why would you want it all together into one sequence? If you have it all together into one sequence, it'll make three different products that eventually it pulls together and nixes out. But it's then just one injection that you have to do. So the methodology, like the development of this for future things, just becomes a whole lot heckin' easier. And so that's one of, I think, the really cool takeaways to this. The source of the green. So the green is called GFP. That is a sequence from jellyfish. They take that sequence and they manually add it in. In theory, you could... So in theory, this part here, so that second part of the green strand, the longer one, that's the one that you're changing out. That's going to be specific to whatever RNA that you're really interested in. The upper part is the green part. And so here's the way of telling it too. So you can see right here in panel D, when that gene is turned on and you're looking at this leaf, you see presence of green. And that's your big takeaway. Now here, this one, that's this construct here. This is a interesting, this is a control experiment. It's called GFP dead. GFP dead is sequence-wise the same as regular green fluorescent protein, except it doesn't glow. The reason it doesn't glow is because it's been intentionally broken at one of the protein sites. So it has structurally, it looks the same as regular GFP, it just doesn't glow. And the reason you want to test that is to make sure that you are not messing up the actual gene itself, right? So the what you're doing now is that does having glow ruin the gene's activity? You shouldn't see any difference. So this is important for later on experiments, but also visually it's GFP dead, so you shouldn't see any green glowing, and sure enough, we don't see any green glowing. Where in the plant's life cycle do this? So let's go to the methods. I want to show you their words. Yeah, so in like the methods of the paper is probably where they're gonna put it. Can't remember if it was like any zone. Let's go to the methodology though. Plant materials, constructs, grain. Here we go. Leaves of 5 to 6 week old plants. So Jay, they were using tobacco plants. The reason they were using tobacco plants is because those are commonly utilized in the plant world that are genetically manipulatable. So they were doing in the plants already like sprouted. That is a powerful thing to test. Because if it's already sprouted, right, then you can really show that it's not just in the seed state that you have to do something, but in like other states as well. You can make sure that you can test and ask the question of, along in this plant's development is also done. Yeah, could it be done in older? Maybe. I don't know if it would be a reason why you couldn't. It could be done. I don't know. My thinking is no. My thinking is that this is an optimal time because they're already growing out the leaves. But yeah, I would guess that they do. They also put it into Arabidopsis, which is a weed. That they use a different type. They use what is called, so they use a virus to deliver whatever molecule that you and gene change that you want to deliver into tobacco. Into Arabidopsis, they use this floral dip method. So the floral dip method is literally what it sounds like. You do a tear and take off a piece of the plant and you dip it into a solution that has your genetic modifiers in it that you want, and then it soaks it up and it's genetically altered. Like that is why Arabidopsis was used for so long, is because it is so amenable to being altered in terms of its genetics. I mean it's really, really cool, right? That it's so easy to actively mess with. And that's why historically it's been utilized. Now, a lot of times nowadays, like we said, they use rice plants, but you can still use all of the genetics that have been identified in Arabidopsis as a means to either as a primary way to check what you've been doing, like let me do a sanity check and make sure what I'm doing is on the right path because it's quick and easy, or as like a supplemental, here I tested it in rice and I tested it in this and it got me the same result, which is always a good thing to do. Yeah, so here's the reagents. So they have a viral delivery mechanism, they have the transformation of the dip, the cell permeating peptide, they also use nanoparticles, and they put it into different plant-based matter, and that gives you your output. The other thing of doing 2-node-J is that you can put in CRISPR molecules. So the genetic engineering that chops out a gene of interest and puts something else in, that too you can put into the liquid. And the Arabidopsis cells just like readily absorb it. There is something fundamental about the Arabidopsis plant that makes it conducive to this kind of methodology. And it's not just that it happens in its cells, it happens then to all of its cells. That's how you can establish a genetic line too. So like you can then propagate it further, and all of the offspring or the F1 also will have, yeah, exactly. It's part of the plant. The solution is also important, but it's more about the plant in this case. If the plant didn't work the way it does, you would never get this system looking the way it does. There we go. So here's again like another example of how this system works. This is an alternative way of doing. So what they do in this instance, they take this green here, makes green fluorescence protein. They've split it into two. They split it into two components. It will not glow green unless it is bound to one another. The way they get it to bind to each other is by putting in this ribozyme fragment. Fragment 1, fragment 2. If both are in the mixture, they come together. Cool? And then here is the additional part. This is what made it specific. To one ribosome fragment, they added a guide RNA. To the other ribosome fragment, they added a second guide RNA. When put together, this matched a sequence. That sequence that was matched is what gene it bound to. So that little pink part is what gave it specificity. God bless you, Cricket. This gives it specificity to a gene. So this pink region you can switch out to be whatever you want. It doesn't matter because what you end up doing is green connects back with green. Does that make sense, Glued? It's just a really, really cool method, right? You're splitting the GFP sequence. If it's split, it doesn't glow. It will only glow if you bring it together. And the way that you bring it together is by adding on these ribosome components. 1 and 2 have to be attached to it. Does the RNA? It does. Yep. It still makes it, Jay. So the RNA still gets... So the RNA is made into protein after this. All this is doing, if it binds to the site, then this green is brought over. So here's the catch of it too, right? In the first figure I showed you, Jay, when the ribosome pieces came together, they turned on the green. Here, they only turn on the green if they're pulled together and bound at the same site. Because that's the way they end up connecting, is that there isn't actually a... Let me zoom in some more. There isn't actually a physical bridge formed between the blue regions. Right, the blue regions are still technically two different pieces here. And the only way that they interact is if this pink region interacts with this pink region. That gives it that specificity. Then this green can be brought to this green, and then that RNA will be turned on into green fluorescent protein. And then this target, the gene that you're monitoring, will also be expressed. What this doesn't tell you, Jay, is amount, right? So it will tell you amount of RNA of instances that are bound. So if there's more transcript target, you'll have more of this take place, which means you'll have more green. But that is only monitoring at the RNA level. It does not tell you if at the protein level it's very, very high or not. That's an important distinction here. Pink region only works if it's connected to the specific RNA sequence. Exactly. And it's both regions have to connect. Having one alone does not work. And the reason that's important, Jay, is what if this guide RNA here matches somewhere else in the genome? It should be designed in a way that it doesn't. But let's say that there is a region that we don't have sequenced very well, and this thing might bind there, right? That would suck if it turned green from just the one binding. And the way that you counteract that is now that you have that secondary one that also needs to bind, it's not going to work, right? Like in terms of like winning an award. But they demonstrated this, like we said, in the tobacco plant. And actually what was really cool, Jay, is in the tobacco plant, they used it for two things. One, they used this methodology to see if the tobacco plant had certain genes that were going up and down, right? Which is the point of what you're doing here. The other thing that they used it for, Jay, is they made that pink guide region specific to a virus. So Jay, if you saw green, it's because a virus infected that plant, right? So that, because the way it works is the same. The RNA transcript was just viral. So if you infect the plant with a virus that might, be detrimental to your crops, the way that you can tell that ahead of time is that this construct, the upper one, the green, blue, and the pink, don't bind to an RNA target that's native to the plant. They'll bind to the virus because the virus also uses RNA. So it's the same principle. It's now you're just measuring for external things coming in, right? So imagine now you can be looking at a field of crops and you could figure out, okay, is there something that's here that could be causing some kind of level of infection my crops that I need to look out for? And that is like a really, really powerful take too, of like, presumably you'd be able to get and figure out before the virus actually spreads, which is really, really cool, right? So like the ecosystem and like the crops could be better protected. They also, like we said, we put it in Arabidopsis, where they just looked at genes on and off. And in the Arabidopsis plant, they looked at a couple of different zones. They looked at leaves, roots, flowers, and stem. Oh yeah, look, here it is in a leaf. Okay, this is just a cool photo. This is not the Arabidopsis. This is the infected tobacco plant. So that's a zone of activity here. So like, look, you can see this, right? The red is the whole leaf, right? And then those green zones are gene activity. And so here it's a really clever experimental trick that they use to prove that it works. The positive control here is they put in a second sensor, a sensor that just works in response to a certain stimulus. So they did a droplet of a chemical onto a side of the leaf, and so you would predict that it should respond, and it does. And then they did a viral infection that is usually wins on this side, and then a viral infection here that is not specific to the plant. So this non-specific one doesn't do anything, but this virus is detected at that site. Hey, y'all, you can see this! And to be clear, you need a certain wavelength of light to check this. This isn't just if you walk in your field, you're going to see a glowing leaf. You do need certain technology at play, but it still gives you like a little alarm if there's something happening to your plants. Do y'all see here the roots? So that glow is in the root of the plant and it's showing activity. And again, activity here is just measured by seeing green. And so again, they had to prove that it works in a lot of different tissue. Otherwise, why do we care? I mean, it would still be cool, I think, if it only worked in a leaf, but showing that it can work in all parts of the plant makes it an even more powerful tool. Especially if you're doing something like monitoring for disease states or monitoring for stress, right? Different plant pieces will tell you about the stress at different times. So it's, I think there could be a lot more information to be gathered from it. So I don't know, what do y'all think? That's a pretty cool paper, right? This isn't the only example of a biosensor, like we said. It's the only one.
What is a biosensor?
In plants. I thought y'all might like this paper, and the reason I picked it was because it is a really cool piece of technology. It's in plants, and it is a first time having been done in this. So it can, Shroom, yes, it can. The way the authors use this was they made it so that you can have it injected into the seeds, so that it's passed on to the next generation. Or let's say, Shroom, you don't want to have to go through that for a lot of different genes. Let's say, Shroom, the diseases that your crops experience change yearly. Let's say every 5 years there's a pulse of disease X and disease Y is the other years. Instead of making seeds for each of those years that are specific to that disease, you have a biosensor that you can droplet from a beaker onto the leaves, and they integrate and they're able to say, like, give you the output of that, right? That I feel like is an especially powerful tool. Does that make sense?